Our final session at Grand Bowling B is Meta Research Innovation, Tackling the Replication Crisis for More Robust Future. Scientific research is a cornerstone of progress, yet issues with reproducibility challenge its credibility. Addressing the replication crisis is crucial for maintaining public trust and ensuring evidence-based policymaking. This session will explore innovative methodologies, open science practices, and systematic reforms aimed at enhancing research integrity and reproducibility across various fields. I'll continue to serve as a moderator for this session. My name is Kyun Lee, Associate Professor of Creative Technology Management at Yonsei University. But first, please join me opening the floor for a video lecture from John Ioannidis, Professor of Medicine at the Stanford Prevention Research Center, Stanford University School of Medicine.
Thank you so much. I'm ready whenever you're ready. So I guess I would need to share my slides and thank you for the very kind invitation to join you, and apologies that I cannot be there in person. I will share some thoughts on reproducibility in the AI era. There's many challenges and opportunities. AI increases computational and overall complexity. In particular, large language models may even defy strict reproducibility because they're continuously updated in terms of their background knowledge. AI may also lead to massive transformations of scientific discovery, appraisal, but also misconduct. And in this regard, it could be both helpful and harmful. We tend to think often of AI as a black box because of its computational complexity and the way that very complex algorithms could be hidden in a black box. However, this may not necessarily be the case. For most AI tools, we have ways to enhance the reproducibility of computational methods. This is an early set of guidelines that we had released in Science in 2016. I think they still remain relevant in trying to define different levels of reproducibility that we can hope to achieve in different studies and that could be adopted by different journals. AI may shift the scientific literature and move science also outside the scientific literature to a larger extent than we're customarily used to. For example, Stanford, I believe, has the highest computational capacity than any other university worldwide, but still this is only 1% or less of the computational capacity of Alphabet, Meta, Microsoft, or Apple. Most AI research in this way may grow outside the published scientific literature, and it may or may not be open source. So we may be witnessing a situation where some very influential work is not even minimally documented within the classic scientific literature. There's a trade-off between transparency and complexity. If we want to present a linear regression, this is very easy to do. We can just record the values, the coefficients, and the constant, and that would be sufficient to get a sense of what that regression is. As we move to more complex models, though, the transparency with the same amount of effort would become less as the complexity increases. And in that graph here, we have deep neural network algorithms being at the bottom of transparency and at the top of complexity. This doesn't mean, nevertheless, that we cannot achieve transparency if we have the right workflows in place and we use proper documentation of code, of software dependencies, what kind of models exactly we specify and what frameworks are being adopted. This is from a paper that we published in Nature where we give specific instructions and guidance on what resources one could use to make sure that even the most complex type of artificial intelligence work could still be transparently and reproducibly reported in the literature. We already see stunning successes with some of the new tools that we have available. We had a Nobel Prize awarded a few months ago based practically on some of this tremendous advancement in the ability to have accurate protein structure prediction with new tools that would be unimaginable in their absence. We also have a growing literature of thousands and hopefully soon millions of papers that mostly nevertheless describe incremental benefits, most of those in hybrid uses of artificial intelligence tools along with the human factor that I think will not be something that we will replace in most of the applications of these new tools. It was not long ago that we were using these very inefficient approaches to ask questions and try to get answers. For example, in the medical field, that their training was very limited and their performance was also modest. Currently, we see that in many areas of knowledge, including medicine, some of the AI tools that we use seem to be performing amazingly well. They get high scores in medical exams. We still don't know whether this is entirely valid or to some extent it may represent leakage of information, as these models can train on previous questions or similar questions that have been available in the vast sea and cloud of online information. We also start seeing a very large number of randomized trials that try to assess specific artificial intelligence tools. For example, this is a systematic review that we published several months ago, summarizing randomized controlled trials that have been published that evaluate artificial intelligence specifically in clinical practice. We have a large number, but we have a vastly larger number of studies that are still unpublished. Some of them we know about their existence because they're registered, and others probably we don't even know that they're happening at the moment. Most of the published results are positive and very promising, but again, if we only have a handful of trials versus a much larger universe of investigations that are not available yet, we don't know exactly what the real value of these different approaches might be. And obviously, our ability to think reproducibly about this literature may be limited. LLMs may also help in evidence appraisal. Evidence appraisal is central in the era of reproducibility revolution, and there's many such efforts that try to appraise to what extent this is feasible. This is from a paper that we published recently in the Journal of Clinical Epidemiology, benchmarking human AI collaboration for common evidence appraisal tools. They seem to perform pretty decent in terms of getting information that would be relevant to Prisma or Amstar. They were not that good in trying to get information that would be relevant to assessment of pragmatism of randomized trials. And the other challenge is that they can cost a lot. So if you want to make evidence appraisals in large scale and try to tackle all these currently hundreds of millions of papers that are the official scientific literature, that would be quite a task, and it might be quite expensive, as it might be for other types of efforts where AI may be used to try to assess different reproducibility aspects. Clearly, we have the potential to make great progress, both in science in general and in medicine using these new tools. We also have many limitations and challenges, including the uncertain performance in many cases, the possibility of spreading misinformation that would be very damaging for any sense of reproducibility, inequalities and other societal impact that are generated by the unequal availability of these technologies, the impact on the scientific ecosystem that is largely unknown, ethical issues and broader issues that could go all the way to existential threats, as some of the gurus in the field have claimed, although I think that this is highly speculative. LLMs may also create a massive fraudulent scientific literature that obviously will make our task far worse. Many publishers have started using AI tools to detect AI LLM tools being used in creating paper mill products, and Wiley, for example, has announced that 1 out of 7 submissions to its journals seem to be fake paper mill products that are just generated massively with current AI tools. There are some features that may suggest that this may be happening, and this is a partial list of when one should start worrying about a paper being fake and created by AI. None of them is waterproof. All of them have limitations. All of them have some sensitivity and some specificity. If you have many of these indicators, perhaps one should worry more, but we don't really know in many cases whether a paper is true or fake. And I think that we run the risk that in the future, distinguishing a fake from a true paper may be even more difficult. For those who are interested to see progress in the field of peer review and scientific publication, which is a very rapidly changing domain, please join us in Chicago in September. We have the International Congress on Peer Review and Scientific Publication, and there will be a lot of very interesting work and interesting discussions happening, I believe. To conclude, hope or hype, I think more of the latter, but there's also hope. Open or closed, unfortunately more of the latter, but things might change. Benefit or risk, probably small or modest for both. I think that there's a lot of excitement, but as in any other technology, probably our initial expectations outpace reality. So we have to watch and see what happens also with AI and LLMs and how they affect reproducibility. Predictable? Not really. I may be completely wrong in everything that I told you today, so please watch out, because things may change very quickly. And finally, is that all ethical? I think it's really up to us. Hopefully, it is still up to us. Thank you so much.
Thank you, Professor John Ioannidis, for your insightful remarks. Now, we're honored to have with us in person today, Jae Shin, Professor in the Department of Pediatrics at Yonsei University College of Medicine, Hyungbom Henry Kim, Associate Professor in the Department of Pharmacology at Yonsei University College of Medicine, and Sarah Soyeon Oh, Research Professor at the Institute for Global Engagement and Empowerment, Yonsei University. Each one of them will give us a lecture on their fields of expertise. Professor Shin?
Thank you for your kind introduction. So I would like to share the concept of meta-research and its role to improve the reproducibility in AI research. So research is also called research on research and science of science. So reflect the previous research practice and find the solutions to improve it. So it fights with bad science, and it covers all fields of scientific research. Professor John Ioannidis has ever said, science is the best thing that has happened to human beings, but we can do it better. So there are many monitoring systems. For example, Institutional Review Board for research on humans or animals, or data safety monitoring system for randomized controlled trial. How about monitoring of research itself? Have you ever thought of this? So meta-research do this role. So meta-research reflect the previous research practices and check the biases and in anything. So it's methods, reporting, reproducibility, and evaluation and incentives. So there has been many movement of meta-research, and so for example, several centers have been launched during the past 10 years. And as Professor John Ioannidis has ever mentioned, peer review congress was the history of about 30 years, and it is held with the editors of major journals. Also, meta-research impacted on the changes in animal guidelines, not only in United Kingdom, but also NIH. And also, it affected to the journal editors, and ICMJ adopted the Equator Network. And PLOS Biology introduced the section of meta-research. And there are many reproducibility efforts by United Kingdom and Open Science. So last year, there was Nature News that introduced United Kingdom launched meta-research unit at the governmental level. This is the first. So Nature introduced this as a very big discussion, and so launched the meta-research unit. Because there have many waste in research. Many fundings are very many waste and not reproducible. So there has been efforts by UK reproducibility effort, then this was a creation of this kind of center at the governmental level, I think. So I and the professors of Underwood International College launched the Severance Underwood Meta Research Center together. So we do this like activity here. So we evaluated all the research area. So method, so this is the phase of designing and conducting research, and as you know, the rigorous research methods are the heart of a good science, and they are having many flaws and biases in the research. So meta-research guide the research to be the more credible, yeah. And this is one example in biases in epidemiologic studies. This is the Now, for example, if you see the result of observational study, the effect size is very exaggerated, but RCT, the effect size is somewhat attenuated, but still they have the bias. So, but if we only selected the trials with low risk of bias, it's totally non-significant. So suggesting many biases in not only epidemiologic studies, but also RCTs. So there are some papers on the biases in AI research too. So this is the neuroimaging, artificial intelligence studies, but there are many so high risk of biases and poor reporting quality. So it's very difficult to conclude, but they inspected the many problems in the AI research. So regarding reporting, it's the phase of communicating the research. So the Ottawa University, there is a Center for Journalism, and David Moore made many guidelines in the medical scientific field. So we have ever discussed on this matter, and his group is, so he's very enthusiastic for the open science and reporting guideline. So there is an Equator Network. So this was first conference was held in Oxford 2006, and after that there have been many. This Equator Network adopted many guidelines. For example, randomized control trial, consort guideline, and observational studies, STROBE guideline, or animal studies, ARRIVE guideline. Currently, about 500 reporting guidelines are here. So there is also one guideline on artificial intelligence in intervention-related one, the Consort AI guideline was adopted in Equator Network, and it was introduced in Nature Medicine, BMJ, and Lancet Digital Health. Also, Spirit AI was also adopted and introduced in Nature Medicine et al. But this study shows the concordance of randomized control trial for artificial intelligence intervention with Consort AI reporting guideline. But I think the education is the problem. So many RCTs has not adopted this AI guideline, and so only just a few adopted some component of it. So it should be improved in the future. Regarding reproducibility, it's the phase of verifying research, and many research has not been reproducible, and this is caused research waste. So meta-research emerged as the reaction to the replication crisis in waste in research. So irreproducibility is reported to be until 90%, and generally 85% of the biomedical research is waste. This was discussed with, especially with the Oxford professors. And so Nature investigated the survey, so to the 1,500 scientists for the reproducibility. It's about 9 years ago. So they felt that 90% said, yes, there is a reproducibility crisis. And so next question was, have you ever failed for the irreproducible experiment? So regardless of the field, so not just medicine, but also chemistry, biology, physics, and earth and environment. Also, they felt that there has been a reproducibility crisis. What factors contribute to the irreproducible research is various causes, but selective reporting or pressure to publish, sometimes insufficient mentoring or code unavailable, sometimes a fraud. So this is not just not limited to preclinical studies, in observational study too. So, for example, none of the 52 predictions from observational studies were confirmed in randomized clinical trial. That's surprising. So many journals began to discuss on the reproducibility and waste of research, including Lancet, Nature, and Science. So there was many discussions. So major stakeholders developed 20 question criteria for the research questions for the transparency, replicability, ethics, and effectiveness for the machine learning and artificial intelligence research for patient benefit. So there are many sections, 20 sections, so inception, like that, reproducibility, application. In these 20 questions for the reproducibility of research, there was 3 questions. So data accessibility and open science, for example, transparency, like that. So evaluation is one component of a phase of evaluating research. So scientific research is constantly evaluated, and peer review there is also a peer review, but it is not, it is imperfect and inefficient. So there has been many problems in the medical scientific research, and there have been also many concordantly retraction of the papers. So I am Professor John Ioannidis has ever looked at the causes of retraction in the biomedical literature, and as expected, most of them by misconduct and sometimes error. In the artificial intelligence papers, there has been also increasing retraction due to maybe the misconduct and any problems. So the role of meta-research to improve the reproducibility. Meta-research has an important role to identifying the roots of the reproducibility crisis and to address them. So strategies to improve the reproducibility in AI research could include efforts to mitigate the potential biases and recommend pre-registration of studies and transparency and open science, and encouraging larger sample size and replication efforts will be important. Thank you for your attention. Thank you.
Thank you, Professor Shin, for your insightful lecture. Now, Professor Kim Hyung-bum will give us a lecture.
Hello, my name is. Thank you for giving me this exciting opportunity to talk about genome editing and deep learning. And instead of talking about the general thing, I will show some example of how AI is applied in biomedical research in the field of gene editing. So this is a structure of a CRISPR-Cas9. It's composed of a Cas9 protein and guide RNAs, which binds to target DNA in the yellow region in a Watson and Crick base pair. And Cas9 also directly recognizes a specific sequence called PAM, protospective adjacent motifs, which is NGG. So 2 sequences in PAM and 20 sequences in target region protospace, we call it, is matched to the guide sequence in the guide RNA. Then Cas9 is activated and make a double-stranded break in the target DNA. This is a structure of CRISPR. And CRISPR has been used to make a drug to treat sickle cell anemia and beta thalassemia. So the drug is to disrupt the gene that suppress expression of gamma hemoglobin. So in adult state, this alpha and beta hemoglobin are expressed, but sickle cell anemia and the beta thalassemia patient has defect in beta hemoglobin, and gamma hemoglobin is expressed in fetal status, and is not expressed in adult. But in patient with sickle cell anemia and beta thalassemia. If we suppress the expression, if we suppress the factor that which suppress the gamma hemoglobin, then the patient will re-express gamma hemoglobin, and which can work as oxygen carrier together with alpha hemoglobin. So the CRISPR-Cas9 targets the factor BCL11A, which suppresses gamma hemoglobin. Then as the result, gamma hemoglobin is expressed and treat the disease. And nowadays, people are using AI to make a new CRISPR-Cas9 protein using new generative AI models, which is one of the examples. It's Open CRISPR-1 by Prof. Fluent. And prime editing is the latest technology in gene editing. So it's a combination of a guide RNA and it's a combination of a CRISPR-Cas9 with reverse transcriptase. So reverse transcriptase recognizes CRISPR-Cas9 recognizes specific target sequence, and reverse transcriptase make cDNA by the guide of a guide RNA, which is called prime editing guide RNA. But designing of this prime editing guide RNA is very complex. I will show the example. So to correct this one mutation, we can design, we can find the target sequence, roughly 8 target sequence near the intended editing. And there are many different guide RNA can be developed, which is 400 guide RNAs to correct just one mutation, but we didn't know which one is the best. So we evaluate all of them, all of the 400 guide RNAs over 4 months to find the best working guide RNA, which is shown in ID88, which shows very highly acting guide RNA. But majority of them was low, so the low efficiency, lower than 5%. So but it took us 4 months to identify that guide RNA by experiment. But in the future, we will not make new prime editors for many patients. In that case, it would take maybe years or decades for hundreds or thousands of patients. So we developed the high throughput method to measure the guide RNA efficiencies. We started from the CRISPR-Cas12a or CRISPR-CPF1. But based on this high throughput method, we collected a large scale of data, which was used to make a deep learning model that called deep CPF1, which predicts activity of CPF1 in cells. Then we expanded this approach to high throughput evaluation and deep learning to Cas9 and base editor and prime editors. And in case of prime editor was so complex, so we analyzed hundreds of thousands of pegRNA efficiencies, and the resulting large data set was used to make a deep learning model called Deep Prime. And x-axis represents primary efficiency measured by experiment. Y-axis represents primary efficiency predicted by our deep learning model. So we were able to almost accurately predict primary efficiency. So the Pearson correlation coefficient is 0.84. But if we repeat the experiment, usually the Pearson correlation coefficient is 0.89 or 0.9. So it's a very close to the real world experiment. And when we used this new model, we were able to increase the primary efficiency up to 30 to 50%. So using this model is open to the biomedical scientists. It's open to everybody as a web tool. And And the reproducibility of this work is very, I think, important. But we opened the training data to the web, so I think the data hope that it will be reproducible. So if you Google and if you click the D prime, and the third one, not the first one, third one is our deep learning model. Thank you for your attention.
Thank you, Professor Kim, for incredible talk on genome research and replication. Now, for Professor Sarah Soyeon Oh, our research professor at the Institute for Global Engagement and Empowerment, will give us a lecture.
Hi, everyone. It's a huge honor to be speaking here today at GEEF 2025, and thank you so much to Professor Lee for organizing this session, someone I respect really much as a researcher and mentor. I think that it's a huge honor, especially because I have a background in preventive medicine and public health. And basically, John Ioannidis has famously said that the failure to win the war on cancer right now is because too many scientific discoveries are wrong. And today I'll be speaking about a research project that I did on the predictors of immediate and delayed onset of PTSS in the aftermath of a natural disaster, a machine learning approach. And basically, I want to show everyone how research on research in my field of public health is being used and sometimes not used in the United States in a good way. And I actually wrote and published this paper when I was a postdoc at the Harvard T.H. Chan School of Public Health last year, and it's under review. We worked in collaboration with PhD students from MIT, EECS, and universities in Japan. So I'd like to give a shout out to my colleagues, especially Professor Ichiro Kawachi from the Department of Social and Behavioral Sciences at the Harvard T.H. Chan School of Public Health, who was the corresponding author for this project, and my colleagues at MIT. So basically, for this publication, we looked at both predictors of immediate and delayed PTSS onset following a natural disaster, i.e. the 2011 Great East Japan earthquake. And I think this is very relevant to GEEF 2025 because it is about the environment, and we are starting to look at how natural disasters affect the mental and physical health of people, both in the immediate and long term. So delayed onset of PTSS was analyzed using machine learning techniques because we really wanted to focus on hidden risk factors that have not been analyzed before. And at Harvard, it's highly recommended right now for all postdocs in the field of preventive medicine or public health, epidemiology, biostatistics, to collaborate with computer scientists at MIT and really try to incorporate machine learning techniques into their research. Basically, this is because initially we've been using regression techniques using programs like SAS, SPSS, R, and the traditional methods that we've been using are now out of date. So the scholars at MIT highly recommend that we also compare traditional results of our research to newer results using Python or R and machine learning techniques. I think it's also important to note that the data set that we've been using in this study is very unique. So basically, my mentor, Professor Ichiro Kawachi, visited Japan's Iwanuma district before there was an earthquake, and he collected data on all of the older adults there. And basically, a few months after he collected this data set, there was a very large natural disaster that basically inebriated that city. And he was able to collect data many years following that to kind of track in the longitudinal sense of the word how these people's physical and mental health are affected by a natural disaster. So to begin with this study and an explanation of the methods we used, I'd like to show that we used a number of computational analyses techniques, including traditional methods like logistic regression, which is very commonly used in epidemiology studies in my area, but also more machine learning-based techniques like boosting models and random forest to analyze the aftermath of a natural earthquake disaster. So let's first see for the audience who I think may not have a public health related background, what post-traumatic stress symptoms are in general. Some people may be familiar with the concept of PTSD, post-traumatic stress disorder, and this is basically a disorder that is when people have difficulty getting over or recovering from an event that is very traumatic. It could be a car accident, it could be a natural disaster, or it could be more unique like kidnapping. And relative to post-traumatic stress disorder, we also have post-traumatic stress symptoms. The definition is that someone might experience severe shock or trauma following a potentially traumatic event, and it's a precursor to PTSD. Usually, symptoms can include nightmares, flashbacks, anger, and irritability, to more serious things like suicidal behavior. And basically, when we were looking at the long-term secondary effects, we wanted to do this because the data in the existing body of literature told us that throughout the years, in various circumstances, PTSD prevalence after an event like a natural disaster or earthquake results in decreases by about 37% following the 1 to 3 months of an event to less than 10% at 10 years. But because this is longitudinal and so much time passes after an event occurs, we wanted to know if these studies were accurate and, most importantly, replicable and transparent. What we did know is that in the existing body of literature, we'd seen previously that even offspring intergenerationally could be affected by victims of a natural disaster. For example, children with mothers that have PTSD might have increased risk of PTSD and anxiety themselves. However, there were problems here, and some questions were still not being answered. First of all, there was a paucity of research on delays themselves, and often levels of delayed PTSS onset differed among smaller studies relative to larger studies, and we have seen the issue of sample size affecting the accuracy of results here. We keep speaking about biases also in this session, and this was highly prevalent in our study too. I'm not going to pretend that our study did not have flaws when it comes to such unique small sample study data, especially within studies that are related to the environment and more longitudinal in nature. There were higher levels of attrition with the data that we used because, number one, our patients were very traumatized after a natural disaster, and number two, this was being tracked over a very long period of time, over 10 years actually to be more specific, and our participants were mainly older adults, meaning some were no longer willing to participate in the study over time. So these were the biases. There was attrition, selective participation, and while larger sample sizes are able to overcome related biases to attrition or selective participation, I'd like to emphasize that because of the unique style of data that was used in the study, we had trouble imputing the data that was missing from our study because we couldn't identify patterns in the data set and predict what patterns might emerge in the future. So I think this session is very interesting and meaningful as a public health epidemiologist because we have a computer scientist, Professor Gion Lee, we have a medical doctor, Professor Shin, and a pharmacologist, Professor Kim, and then a public health person, i.e. me, who is trying to use machine learning to do some kind of preventative medicine-style research that is transparent and reproducible. So I feel like meta-research, i.e. research on research, is going to get so much easier and simpler as more scholars practice transdisciplinary research and incorporate machine learning and AI to produce research that is more flexible, transparent, and has fewer assumptions comparable to other studies. So in the second part of my study, I'd like to actually show you what we did, which is based on machine learning. And I think that many people here would probably be unfamiliar with the concept of machine learning itself, like I was once as well. So basically, machine learning is a tool that uses algorithms to analyze large amounts of data to help improve decision making. At least this is what's happening in the public health field. So we can identify patterns, generate insights, and make predictions that ultimately help medical professionals identify new diseases and predict treatment outcomes. I feel like machine learning is something that in healthcare can help us in so many ways. We can improve diagnostics, advance treatment. We know that lots of systematic reviews and meta-analyses are helping us to try and group research findings and tell a story, some kind of consensus about diagnostics, treatments, and or related tasks. Machine learning, I think, can be used in this process because, as I will show you, machines can reproduce our studies hundreds, if not thousands of times and make sure that they are accurate if done in the right way. So in existing systematic reviews of PTSS, we've seen that around 84% of studies have relatively high accuracy rates in predicting short-term PTSS and PTSD. For example, there are a lot of studies on the 2013 Sichuan earthquake study, and when there is a combination of information on gender, age, being employed or unemployed, and physical conditions, we can quite accurately predict which individuals are at high risk of suffering mentally after there is a natural disaster in their lives. In our study, we had a very unique data set, as I aforementioned. So we used data from the Iwanuma study, which is an amazing data set that basically has data on residents aged 65 and above in the Iwanuma region of Japan. So 7 months following the Great East Japan earthquake in 2010, we were able to gather data not only before the earthquake occurred, but after the earthquake occurred to survey survivors in the district from 2013 to currently. So we've had data immediately before and for a long time after a real-life natural disaster in this study. These were the variables, and you can see they were socio-demographic, lifestyle, or earthquake-related. And with the earthquake-related variables, we had individuals being asked about whether they'd lost any loved ones, pets, how much housing damage they were experiencing. We also had data on post-earthquake health-related variables, so we could see how their health deteriorated or changed 2 years, 5 years, 10 years after a natural disaster. In our study, because we looked at both immediate and delayed PTSS onset, we made sure that a score of 6 or above in the PTSS scale, so immediate PTSS, was defined differently from delayed PTSS. This was defined in our study as 2.5 years or less after the earthquake, and anyone who did not have PTSS in this period but had signs of PTSS after in 2016 or 2019 following the earthquake were reported as individuals with delayed PTSS. And as you can see, we did incorporate traditional programming methods like SAS to code this. I'd like to show the audience why I personally think that machine learning can be used so interestingly in public health and preventive medicine. Basically, my brilliant colleagues at MIT taught me that machine learning studies can split and test and train data in multiple ways so that the split data is trained on subsets and evaluated for performance accuracy of each other. In other words, by splitting our data set, we could avoid problems with overfitting so that the model is not only trained on the training set, but also tested on the test set, validated on a validation set if necessary, and we can assess how a data set performs overall. One part of our data set can be used to test our hypothesis, while another part of the data set tests replicability, and exact same tests to ensure that our model would be effective in real-world scenarios. There's also something very interesting called hyperparameter tuning that sometimes happens in machine learning research and sometimes does not happen. And I think that every single machine learning study in public health actually needs to be very careful and make sure that they're hyperparameter tuning their data correctly and bootstrapping their data. So basically in ML, a data set can be sampled multiple times by AI, and replacement data sets of the sample size that is the same as the original can be calculated so that we can figure out if our desired statistic is correct. And this process can be bootstrapped or basically repeated up to 1,000 times for validation. And we can use these values to find a probability, i.e. the likelihood of something happening. In our data set, when we used a normal traditional style of logistic regression to analyze and predict PTSS, we found the expected ideas that were emerging in the data values. For example, a person who had separated from their partner was 3.38 times more likely to develop immediate PTSS, and someone living alone was about 2.04 times more likely to develop delayed PTSS. We also saw that earthquake-related loss, such as losing a relative or a friend, greatly exacerbated the odds of immediate onset and soothed down for delayed onset. However, loss of relatives or friends, human relations was immediate, whereas the loss of a pet was a bit delayed. Housing damage was also very highly associated with immediate effects. We can also see the health-related metrics. So post-earthquake health was associated with very immediate effects of stroke, heart disease, diabetes, and fractures. And interestingly, in our machine learning analyses, we found that our data set, when using different techniques, was kind of accurate, not too accurate. but kind of accurate. So our logistic regression model was able to predict outcomes by up to 74%, whereas our gradient boosting models, our light GBM model, was about 62% in accuracy. So basically our data set was able to predict to some degree the individuals that would develop PTSS in about 10 years' time. We were also able to see which factors were more important in our model, and we could see which variables in our data set are compulsory for the prediction to occur correctly. So basically what we noticed is that there are different findings in immediate and delayed PTSS onset in our research field, and our data set found it a bit difficult to replicate our findings with pristine accuracy. We also could see that factors like housing damage were associated with immediate PTSS, but other factors like pet loss would result in delayed PTSS, although our accuracy was only around 60 to 70%. We've seen that machine learning is interesting when it's used as an aid in public health research. It can't be a full-on substitute. Previous studies have found that PTSD can be predicted by up to 75% accuracy when we have data on the timing that a person has stayed in an emergency unit or the kind of medical treatment that they've received, but we did not have this style of data. I feel like in public health, we still have a very, very long way to go because many machine learning publications, as the panelists in the session have emphasized, are still very unclear. And there's eventually going to be a machine learning crisis in scientific research because, first of all, if ML is not used as a supplement but main answer to research questions, sometimes algorithms are going to be fixated on noise that cannot be reproduced by other studies. And there's just going to be so many bias problems if a researcher is not knowledgeable of what they're doing. And there can be always problems of overgeneralization, difficulties overcoming attrition depending on the data, and lack of knowledge about the data itself. So today, I think I'm going to start by sharing my GitHub. I was very impressed with Professor Kim when he just revealed his data as well to the public. So basically, I think that it's important we all take the first step in making what we've done in our research very public to the general audience. So this is my GitHub with the code that I used for this data set. I think that it shouldn't be embarrassing for a researcher to receive feedback from other people, especially people from different fields. And researchers shouldn't be so wrong to the extent, as Professor mentioned, that fraud is happening. But I also think there needs to be a better platform or progress checking methodology where researchers are able to receive feedback from other researchers in different fields and openly speak about whether their code is correct or whether a data set is difficult. We don't reprimand people for making mistakes when they speak a new language for the first time, and I think we need to work on some kind of way that allows researchers to open up their data and code and receive feedback from other researchers in various fields who are experts to check their work before they officially publish their findings and influence the policy-making progress and lives. Finally, to conclude, I'd like to share that as a maternal and child health epidemiologist, we've been doing what we just did with data on fetal alcohol spectrum disorders now to predict which women who have consumed alcohol during pregnancy will give birth to a baby with developmental disabilities. And this research is so important because with diseases like FASDs, as Professor Shin, who is a pediatrician, would probably agree with me, it's very difficult to diagnose diseases correctly, especially if they're very similar to diseases like autism. And I think that AI is going to be very welcomed in my field when it's started to use correctly. We can see that the prediction rate is going up for this. And I'd like to end with a quote today that basically speaks about the replicability issue. And I'm going to say that because this forum is about bridging divides, there are transdisciplinary research divides that also need to be spoken about. Basically, people in one field present ideas and describe things differently from people in another. For example, terms in epidemiology and biostatistics, i.e. my field, are different in expressing certain terms and ideas from people in the machine learning world. And even though they basically are expressing the same thing, I've noticed that different ideas are communicated in different ways. Finally, I'd like to finish my presentation by reading a quote from clinical imaging. AI will not replace radiologists. Instead, those radiologists who take advantage of AI may ultimately replace those who refuse to accept it. Thank you very much. I hope that the floor is able to communicate with me if they'd like any research collaborations with me in the future.
Hey, thank you very much, Professor Oh, for sharing your unique research. Now it's time for us to have panel discussion. We have about 11 minutes, but should we do this in Korean or? Yeah, okay. Do we have any comments that you want to make, or do you want me to? 제가 질문 드릴까요?
So shall I ask the question? So I was listening to the presentations and I could see that these are quite unique studies. For example, Dr. O
had data that is very unique, and it's not data that you can come across easily.
and Dr. Shin, Professor Shin, carries out meta-research and analysis, and you repeatedly do this, but the samples that you utilize, well, you have been working on leading to a conclusion, and also Professor Kim, well, of course you can repeat what you've been doing, but in terms of the data, well, I'm not sure whether this can be regenerated, but they're very unique. So in order to come towards a general conclusion, that's why we're engaging in meta-research. So what more is required? And also, for deep learning, through deep learning, what kind of new opportunities are given to us? And also, what are some of the risks that we should be aware of? It doesn't matter who speaks first. So let me go first, very briefly, if I may comment on this. So deep learning and machine learning, well, in many cases, well, you don't really publicly disclose the training data. And so I think that that is at the core of the reproduction, because if you don't open up the training data, of course, it's difficult to have that kind of reproducibility. And also, you don't open up the code or the hyperparameter. All of these are not shared or open to the public in many cases. So, nevertheless, the paper is accepted and published. So, that actually hampers and hinders reproducibility. I think that's the biggest reason, the key component that makes reproducibility difficult. So there's no incentive actually to make that information public at this moment, because if we really want science to advance, we should make it public so that other people can utilize that. For example, there should be a platform that makes that possible. And also, of course, there should be a venue or a platform that enables that. But at the same time, there has to be an incentive mechanism. For example, if there is an impact factor, a high impact factor, and you submit a paper, if we have a kind of a process, there's a disincentivized process, perhaps that could have an impact. Yes, if I may add on to that. Now, whether it's the medical field, well, we can see that there is limited reproducibility. And as it was mentioned by Professor Kim right now, the code is not available. And of course, you said that it's because of incentives, for example. And a lot of the journals, for example, they publish the positive findings, for example. So it's not just about AI, but when it comes to the p-value, for example, 0.05, in order to publish, people make efforts, and that is what has led to what we see today. But, well, stakeholders, for example, I mentioned about the 20 questions, and in relation to reproducibility, as you just mentioned, there has to be openness, transparency, and also accessibility to code. I think that that has to be more clarified in order to have trust and reproducibility. I think that there's ongoing talk about this, discourse on this, and the role of meta-research is that I did talk about this in English, but well, the past research practices, whether it's the methodology or reporting, we try to analyze what's been done in the past so that we can go into a better direction and guide people into a better direction. So the reason that we're doing meta-research is that, including Professor Iwanik, and also in the UK, for example, there's a meta-research center, there's a government body, actually, a government unit that's spearheading meta-research. But anyhow, I think that there's a lot of researchers who are not aware of this. Of course, the journals are talking about meta research, but however, there's research done and some people may not be aware. And so I think that through various approaches, I think that there has to be more improvement and there has to be multifaceted efforts that has to be carried out. Professor Oh?
Well, actually, as other professors have mentioned, we don't have the solution for cancer because many research have gone wrong, and I think we have to start with medicine because this is an area where people's lives are on the line, and so medical researchers need to feel that responsibility, and so you need to disclose your codes, and for high impact areas, you need to be incentivized to share your codes openly. And one other thought that I do have is that doesn't mean that we should collect unnecessary medical data, and that should be regulated. Thank you. Thank you very much. On a personal note, I do believe that through institutions and individuals, we need to appreciate people who open their codes and data, and we need to applaud them, and we need to cite them in order to incentivize opening that data. I completely can relate to that. Thank you very much. So we are almost out of time.
Sharing these impactful insights on strengthening the credibility of scientific research. Please give a warm round of applause to our three speakers and Professor Ioannidis, who is not here, but he may hear your claps, okay? So again, big hands to them, yeah.